Thank you very much for having me. I'm excited to be here. And in fact, you're right, it is overdue. I have not yet spoke at Georgia Tech, and I've interacted with lots of you, both academically and socially. So I'm glad to be here to tell you what I do in the basement of Rollins over on the Emory campus. So what I'm here to talk to you about today, and what I'm interested in, is why and how we remember what we remember, both from the psychological perspective and from the neuroscience perspective. And I've picked this picture of my son from last year, our summer vacation at the beach, to kick it off. And that I was going to use this picture as a way to say that to him, it was clear that this was memorable of an episode, because look at how much joy is on his face, and this must have been a worthwhile memory, at least for us to pay all the money to go to the beach. And when preparing this lecture, I showed him this. I said, hey, aren't you impressed? I'm going to talk about you in front of a bunch of people. And I said, what do you remember from this picture? And he said, nothing. So now the example is going to be from my perspective. I'm the one that actually took the picture. And the point is that I remember this relatively well. Am I having a stroke, or is it getting dimmer? Okay. I usually don't get nervous when giving a talk. I don't think it's my, but it is possible I was stroking out on you. I don't know. I guess that I'll have to wiggle occasionally there. All right. So now I'll remember this. But the reason I remembered that was less of the fun splashing in the waves, but that as I saw the memory through his eyes, that to me this was memorable because I experienced lots of emotions about this as a father. That this was both like it was a nice breeze, warm water, pleasant sunset, but also it made me think that I'm a good father. I provide these joyous moments for my son, and I use that memory as part of what psychologists will call their autobiographical narrative. And is that me? Okay. You guys throwing curveballs at me left and right. I thought that the tech part stood for technology. Technical difficulties. Georgia Institute of Technical Difficulties. Right. So there's a reason why both the kind of emotional affect that it conveyed to me, as I feel pride and satisfaction and all of those things wrapped together, and that's part of why I remembered this particular event, even if my son did not. But that, of course, we talk about these moments that we cherish, and we think about those, that how important it is to hold on to those important memories. But I think that we often forget how important it is to not remember some memories, that forgetting is just as important as remembering, because without prioritization of the important memories, you really don't have good memory. And to make that case, I'll show you this picture of this woman, Jill Price, who in a report by Jim McGaw and others was referred to as case AJ. She has since gone on to appear on Oprah Winfrey in 2020 and wrote her own book. But by all accounts, as far as these memory experts could tell, she can remember just about every episodic detail from her life starting in around 1980. And that the way you can assess that is by asking about some things that might intersect with public events, like when did this plane crash, or when did JR get shot, for those people here more my age. And that every time they ask her, what she answers is accurate. Now, we can't assess the extent to which having a bagel and cream cheese for breakfast on September 16th, 1985 is accurate or not, but the things that we can assess were accurate. And that you might think, wow, that would make graduate studies much easier if I could remember everything. But the point here is not how impressive this is, but what of a burden she sees it as. And in fact, she describes, and in this quote, most have called it a gift, but I call it a burden. And it's that she feels besieged by the remembrances of everyday mundane details, that she can't sort out what's important from what's not, and it's everything to her, and that's bad, not good. So then to go further then to ask what is it, what means, and I've been told I'm only allowed to pace on this side of the room because that's what's in the field of view, so I'm not ignoring you, I'm just mugging for the camera. That to ask what is important and what's not, I don't think that we can stop at, the things that tug at our heartstrings as parents or any emotional events. That I think we have to ask it from his perspective as well. And I'm sorry for all of the issues eating. At 11, I didn't think you guys would be eating. Trigger warning, gross stuff coming. This is a tree shrew eating the head of a cricket. And that I think that to ask the question of what's important to this tree shrew might be different from what's important to me as a parent or what's important to you as going about in your day-to-day lives. And the reason I bring this up is that I think that we need to think about memory prioritization not just from the anthropocentric view of humans, but from the perspective that might apply to a broader evolutionary perspective. And more so, what I'll tell you, and this might be a little bit disappointing, that the fundamental memory mechanisms in this guy's like vampire-looking squirrel's brain is pretty similar to yours. So here is a close-up of a cross-section of the hippocampus from several different species, including, this tree shrew here. And what we see here, this is the hippocampus of a tree shrew, mouse, tenrec, and a human. And you can see and I'll just pick the tenrec here, why not? You can see the densely packed cell layers, the pyramidal neurons of the CA fields in the hippocampus, and that goes from the subiculum, CA1, there's a transition area, CA2, CA3, and this other densely packed layer of granule cells in the dentate gyrus. And that to the extent that we've looked at these details in different species, we found pretty much the same thing. And I say we, I mean they, the anatomists. And it might be disappointing to you to know that the anatomy that allows you to remember those cherished memories is not so different from the from the tree shrew, remembering that delicious meal of his cricket's head. Again, apologies to those of you eating sandwiches. So to me, understanding how we remember what we remember, how memories become memorable, the prioritization, I think we have to take into account what might be an explanation that would apply to some extent, at least at the neurobiological level, across the mammalian taxon, because those brain mechanisms are very similar. So the last picture I'll show you to set up why it's important to study memory is this picture of a young gentleman who happens to be me, graduating from high school, and to the left and right are my grandparents. And that I show you this picture not so much to say that this was an important memory to me, though it was, it's to point out the fact that my grandfather was diagnosed with Alzheimer's disease at the time of his death. And if you want to know about the importance of memory, don't ask a patient with Alzheimer's disease, because at some point they become blissfully unaware of their memory condition. Ask the spouse. Ask my grandmother what it felt like to be forgotten by her spouse of 50 years. I think that goes a long way to showing in a simple demonstration how important memories are and what remembering what things are important means to us. So that's the setup for my talk, and what I'll try to do is get through some of that, and I won't get through all of it now that I understand that some of you have classes to get to perhaps as early as noon, but that what I'll try to do is talk about setting up Alzheimer's disease in a rat model to again set the stakes, so to speak, of understanding these mechanisms are important, not only for just understanding how you remember your vacations, but also what goes wrong in disorders that impact memory, like Alzheimer's disease. I'll also talk about then what we know about what marks good memory states in the healthy hippocampus, and I'll then talk about more recent work that we've used to stimulate the amygdala to try to artificially prioritize some memories over others as a window into what makes good memories over bad memories. And then if there is time, which there may not be, I'll talk about work with scout-based EEG recordings from humans doing autobiographical memory retrieval in both emotional and neutral conditions. So I'll jump in with the Alzheimer's disease model, and this is a rat model that Robert Cohen developed when he was at UCLA and Cedars-Sinai and brought here with him to Emory to Atlanta. And it's a rat model, not a mouse model, in which it was a double transgenic. They used the human genes that are from particular humans that are known to come down with adult onset Alzheimer's disease. Early onset Alzheimer's disease. And the two genes are particular the presenilin 1 and the APP, the Swedish variant from those individuals in Sweden that they got it from. So that this rat model is detailed by Robert Cohen in this paper in much detail. And one thing from that paper I'll just show you is that like the neuropathology in human Alzheimer's disease, the neuropathology in this rat model is age dependent. That at some early age, and what we're seeing here is a side view of the rat brain and a zoom in there of the hippocampus. And this here, this dentate gyrus, I think you can see in the red stain for red cell bodies in both at the time point of 6 months and 15 months. And on the 6-month period, you can see that there's not much staining for amyloid deposits, but at 15 months, there's quite a bit. And in fact, as the rat ages to 24 months, it accumulates even more. But just to make the point that this rat model develops the human, a similar type of pathology as in humans in an age-dependent way, including these amyloid plaques and the neurofibrillary tangles that aren't shown here. We wanted to know then where we entered into this then was to try to understand somewhere between these two, and both of these are before there's any neuronal loss or synaptic loss. These are markers of amyloid burden, not necessarily of cell death. But we wanted to know at a very relatively early age what might constitute the hippocampal dysfunction in these rats with the Alzheimer's-like disease. And so the first thing we did with a former graduate student in my lab, Claire Galloway, was to ask at what age do they become show memory deficits. And so here at that around that 6-month age, from 5 to 8 months, these rats, the Alzheimer's rats in gray compared to the wild type in white, showed a memory performance that was relatively normal. And this is a discrimination index of a type of memory. And I'll just say that the 0.5 represents chance performance and 0.66 represents pretty good performance on this type of measure. But then very quickly, by the time they reached 9 to 12 months, the AD rats had developed a profound memory impairment, and the wild type were still performing pretty well. And thus we wanted to know then, not necessarily just at the behavior level, but what was going on in the hippocampus of these rats. And like a lot of what we do in my lab, we use in vivo electrophysiology to interrogate function and dysfunction in brain circuits as they relate to memory. And in this particular picture, you're seeing a rat that we're recording from wires implanted in its brain as it locomotes on this track. In fact, he'll come back and get a little piece of chocolate for every lap. And I'll show you a video of that, of some of these Alzheimer's rats. Oh, sorry. So what we can record from that are spikes from individual units using tetrodes, like several of the folks here at Georgia Tech already do. And here is one way to look at that data. And in this fast forward video of a rat completing laps on the circle track, what I've done is superimpose with red dots a marker on the screen to show you every time this one example neuron in the hippocampus emitted an action potential as the rat was completing these laps. And that I think what you can see is that all of the spikes from this example neuron are occurring in this particular XY location on the track. And that these are well known. They've been well known since John O'Keefe described them and then eventually won the Nobel Prize for their discovery. It's a neuron with a receptive field defined by spatial landmarks that then earned the name a place field, the receptive field, and that the neurons then are called place cells. And that we use these then as a way to look at the hippocampus in these rats to see, is it acting normally? Is it showing normal looking place cells? And here is a more typical way to plot these place fields rather than the video. And looking here across for the wild type animals, you can see that the black path is just showing you where the rat ran. And then now in color-coded with cool colors representing lower firing rates and hot colors representing higher firing rates, you can see that for the wild type rat, these different example neurons from CA1 and from the CA2 and 3 regions are quite circumscribed. They're nice tight place fields representing one place. And that wasn't surprising. That's exactly what we expected from these healthy wild type litter mates. But then in the AD rats, the surprise was that at least for the neurons in the pyramidal neurons in CA1, they also looked perfectly normal. Even at an age, and this was at least at 12 months of age, that these rats we know showed profound spatial memory impairments. The CA1 neurons looked pretty good. And it was only these CA3 and CA2 neurons that showed us kind of spread out and mushy place cells. They weren't firing any one particular place particularly well, thereby kind of robbing that area of the brain information about the location of the rat. And that one of the most classic ways to quantify place cells is with this standard, widely used information theoretic approach measure called spatial information score. And it's just a way to try to ask how much information could you get in kind of bits per spike from a neuron as a rat's locomoting about. And what you're seeing here in gray for the AD and white for the wild type, pretty normal for the CA1 neurons. No difference there between the wild type and AD. But specifically here for the AD, you see a deficit in the CA2-3 pyramidal neurons. And it wasn't necessarily the fact that we saw a deficit, it was that the deficit was specific to one area of the brain. And just to kind of make a cartoon here as I talk, in part because I can, that this is wild type and AD for CA1 and CA3, and it's specifically the CA3 neurons that didn't have these neuronal landmarks, these place fields that were nice and tight to give the rat a sense of where it was, and instead this little cartoon rat is obviously lost in these spatial representations. And that now then, what do we do with this? And I think that the answer, of course, with Alzheimer's disease is don't let the brain get to that state in the first place, that we need to develop a cure or a disease-modifying treatments to impact the trajectory of Alzheimer's disease so that you don't get there in the first place. But that until we have that cure, and even after we have that cure, there's going to be a great need for treatment of symptomology in Alzheimer's disease, because probably for those of you like me who are already in your 40s, even if they came out with a cure, it's probably too late for us. We probably would have needed to be taking that medication now to prevent that trajectory. Yes, ma'am. It's a great question. And so that had the R21 not been an R21 and run out after 2 years, the next step would have been to record from those rats as early as 3, 4, 5 months of age, however early was feasible to do, to ask, were they abnormal all along, or did they develop this dysfunction? And so the answer is, I don't know. The pathology and the memory is age dependent. And so my best guess is that these the dysfunction in the CA2 and 3 pyramidal neurons is also age dependent, but that at this point is a guess. Thanks. And feel free to throw out questions. And I forgot to ask, repeat the question, but yes?
So sorry, Joe, just to piggyback on that, then these data
so I don't have to.
post 9 months?
Yes, the data were from post 9 months. So that then, though, that the question isn't like what to do about Alzheimer's disease. The answer is, cure it. But that it suggests that for the treatment of symptomology, when there's dysfunction in this part of the circuit, it leads to questions about how we might ameliorate that dysfunction. And that the answer is from that of even now, we don't know. We've the hippocampus is one of the most studied brains, one of the most studied brain areas, and of all of neuroscience. I mean, we have our own journal, for God's sake, but that we still don't know exactly what it means for CA3 to be dysfunctional and CA1 to be normal. We don't actually quite understand what the ramifications of that, or even what kind of treatment we would want to bring about. So that now I think that makes the point then that Alzheimer's disease is bad. Don't get Alzheimer's disease. Give money for treatment. But then also we need to understand the healthy brain a bit better so that we can better understand other areas of dysfunction. And so that's now what I'll switch and talk about a bit more, leaving behind Alzheimer's disease and start talking about the hippocampus in healthy rats. And that we do the same thing that we do with the Alzheimer's disease rats, except that now we focus a bit more on the actual memory performance. So this is a similar video of a rat completing laps on a track here, but now I've put objects, junk, novel objects the rat's never seen before, in its path, and it has the opportunity to stop and sniff the roses, so to speak. And I think what you would see on the first path, the rat stopped and momentarily sniffed the objects, but on laps 2 and 3, he ran right by them because they were boring to him, and we infer from the reduction and exploration on subsequent repetitions memory for the objects. And the idea is based on lots of testing in this task and other tasks like it in monkeys and humans that's based on spontaneous preference for novelty. But then we don't stop here, because I think if you looked at this and this similar plotting of from a different example neuron, a red spike every time the neuron emitted an action potential. From here it's unclear, is this neuron its activity correlating with location or with that particular object or some combination of both? I don't think you could tell from that type of short session. So then in fact we record rats running around for an hour or so, lots of different objects and lots of different locations. And here it's there's still no answers from one single video. That we know it's not firing over here, but it seems to be firing for something to do with objects and something to do with locations. And in fact that we don't even stop with this, but then instead go on to recording lots of different neurons simultaneously. And in this video I've color-coded action potentials from different neurons with different colors, that there's potentially a rich opportunity there to interrogate the spike trains to ask, what are the memories? Is there information in the activity in these action potentials about remembering this tin can here versus remembering that hairbrush there? And the answer is, of course, yes, but that's not what I'm going to talk about today. Because the classic approach in in vivo electrophysiology in the hippocampus has been to record from action potentials and try to decode the memory correlates from those spike trains. And it's had lots of success. I mean, it's had Nobel-worthy levels of success. But that there is also another approach, rather than bringing out all the Georgia Tech folks to try to use their machine learning and whatnot to squeeze everything we can from there. And that the other approach, well, let me say, this is not me saying that I think that recording action potentials is a bad thing. In my former career with Howard Eichenbaum, this is everything that I did, right? And that it's the whole reason Emory hired me, so of course I think it's great. And the idea that that I played a small part in, but that was kind of field-wide, is that from asking about the different correlates of neurons, for example, grid cells from the Mosers and the medial entorhinal cortex, that we know that the hippocampus sits at this confluence of spatial and non-spatial information, such that the hippocampus seems to be positioned particularly well to integrate spatial information with non-spatial information, so items and their locations, for example. But what this doesn't tell us, at least not particularly well, is what might be the dynamical system here within the hippocampus, those subregions of the hippocampus that I already told you are conserved from you to a donkey to a tenric to a tree shrew. What's going on within those local connections to mark good memory states compared to bad memory states? And for that, we have taken another complementary approach. And in fact, this is an even older approach than the approach of recording spike trains. It's recording local field potentials from those electrodes rather than the activity of action potentials. And that the local field potentials really just represent the summed activity of lots of EPSPs and IPSPs waxing and waning to lead to these oscillations that you can record in the local field potentials. And that has been of recent efforts, our effort to try to ask, as we record in different subregions of the hippocampus, what might be the oscillatory interactions between those subregions and how might that convey information to us about good memory states versus poor memory states. There we go. So here's just an example to say what I mean. So now instead of trying to get from individual action potentials, record from as it can be one electrode in CA1 and can be one electrode in CA3. And this is an example of one recording from one period of about 4 seconds as a rat approaches and initiates onset here at time 0, exploration of a junk novel object that it's never encountered before. And from this, you can already see, even those of you who are not used to looking at local field potentials, there's a clear oscillation in this that is going at about 8 times a second. That's the classic theta rhythm that you can see in the rat hippocampus, and as technologies have progressed in recording humans as they locomote about, also in humans. And that there's going to be a good correspondence between the theta and CA1 and the theta and simultaneously recorded CA3. And that's, that's true and that's been known for a while. What we wanted to focus on here was this period of novel object exploration. And what I'm going to do now is just zoom in on this section and show it to you kind of superimposed on top of each other for both CA1 and CA3 in red and blue respectively. And you can see that overall theta now for this shorter half-second period. But the point that I wanted to make is that you can also see alignment here at a faster, what is often called gamma oscillation. And we've come to know that there's not necessarily just one type of gamma. There's multiple potential subbands of gamma that have earned names like slow gamma and fast gamma. I'm going to be talking about particularly slow gamma today, something that's about 40 hertz, 40 cycles per second. And so instead of just asking you to look at the squiggles and see that do the squiggles squiggle together, there's a way that we can quantify that. And there's lots of different ways, but the one way that I'll focus on today is a metric called coherence, which not only looks at the phase alignment between two oscillations, but also the covariance and amplitude at any particular frequency band between those two. And so here is one way to plot coherence, again with time being on the x-axis and 0 being the initiation of exploration of a novel object. But now on the y-axis is frequency, and going from 0 to about 100 hertz. And what is color-coded then is coherence, with cooler colors representing lower coherence or lower synchrony between C1 and CA3, and hotter colors representing higher coherence. And that what you can see, and I'm really sorry for those of you, I keep wanting to like give you some over here, but then, that one guy sitting on his couch watching me, hey, one guy, he wouldn't be able to see that. That this almost maxed out thing happening around 8 hertz, that's the theta. And that seems to be going on very strongly, basically when the rat is going on and about and awake and like investigating the world, not just investigating objects. But hopefully what you can see is there's not much here in that gamma range prior to the onset of exploration, but then there's a dramatic increase from about 30 to 55 hertz in that slow gamma range. And that in this particular study that was from before, a graduate student in my lab, John Tripper, found that for CA1 and CA3 coherence specifically, that the extent to which there was an increase in that slow gamma coherence during the exploration of a novel object predicted subsequent memory for that object. And I'm going to dig into it with a more recent study because what John subsequently did after that first study was to record not just from CA1 and CA3, but from all 4 subregions, with my apologies to CA2, that we recorded from dentate gyrus, CA3, CA1, subiculum simultaneously to try to get a bit more about what's going on and the dynamical interactions between all 4 of those subregions. And that this is basically the same plot but with different animals that I showed you before, where 0 is the onset of exploration, showing this increase in the slow gamma oscillations when the rat initiates exploration. And you can see the same thing actually between dentate gyrus and CA3, but in fact it was kind of going on all along, and it goes from a lot to a whole lot rather than not much to a lot. And that isn't a universal necessarily aspect of what's going on in the hippocampus. In fact, there's not a lot of gamma synchrony, at least at this kind of color coding level between CA1 and subiculum. So it's something about a particular aspect of this portion of the circuit. And so to ask more about that, I mean, we have some basic questions like, is this increase in slow gamma oscillation, is it really about forming a memory for the object? That was our hunch, and that's what I'm telling you. But it could be something as simple as, this is what happens when a rat stops. It may be cessation of locomotion rather than forming a memory. It could be the act of exploration itself. There may be 1 or 2 of you that are interested in things like whisking in the audience, and so maybe this is more about a behavioral state of investigating an object rather than the memory state of making a memory for that object. And one way to ask that is to then take what's going on here during sniffing of a novel object and ask again, what does that do to predict subsequent memory? And so here's one version of the task, and John did several, but that we have rats sniffing objects on a brand new junk novel objects that it's never seen before on as it completes laps on the circle track. And that on the very next lap, we repeat them. We actually repeat duplicates just to remove the possibility of scent marking, but we repeat those objects again. And what we should see, and what we did see, is rats reduce their exploration. And that's more to make sure that the rat formed a memory from that initial encounter. And then there were several different conditions just shown here as this could be trial 1, this could be trial 2, 3, 4, different conditions. And so one condition was we took those objects and with duplicates swapped their locations. So if a rat remembered and reinitiated exploration of the toy hamburger, it wasn't a real hamburger, that it would mean that the rat not only remembered the object, but also the object's prior location. Because we put in there control conditions in which the object got repeated, for example, in the exact same place with the same object. And then the other kind of comparison condition was what happens when you put in a brand new novel object. And through something that I won't, the kind of flow chart of how do you say from this that is there good memory for the object and or good memory for the object and its location. It's an inference on what goes on laps 2 and 3 to try to predict back, was this a good memory encoding here at the beginning. So on the next slide, I'm going to show you neural activity from here, but split by conditions determined by those behaviors. And that now what we can see in those memory conditions in this kind of orange-red, when a rat was sniffing a brand new novel object, split by whether it was going to show good memory in the future for both the object and the object's location, or in blue, just the object, or in black, no evidence of memory for either the object or its location. And that this is now shown just as actual power of oscillations across dentate gyrus, CA3, CA1, subiculum, and just the grand average. Because a bit unlike, and it's baseline subtracted, that's why everything's at 0 to emphasize kind of what differs between the conditions. And what differed between the conditions was this slow gamma range, somewhere between 30 and 55 hertz. And it did it mostly for dentate and CA3, somewhat more for CA1 and subiculum, but that one of the most statistically reliable thing was just to take the average slow gamma power in the hippocampus, and that was one of the best predictors of whether or not the rat was going to show us good memory in the future. And so we were expecting complication, and it's, in fact, one of the rare times we got something, a more simple answer, and that answer, at least from this data, was that slow gamma oscillations in the hippocampus, stated kind of generically like that, was a marker of a good memory state. And so that was a nice one of those times where you get clarity rather than asking more questions than answering any. Now, I don't want to say that everything was cut and dry, that the questions of disambiguating memory states here from behavioral states was what a lot of the manuscript focused on, and that it wasn't as if slow gamma is the end-all be-all in the hippocampus. It was just that that particular memory network state correlated best with the inferred memory state of making a good memory for the object in its location. I know I'm talking really fast, so I'll stop. Yes.
Question. So is this effect an emergence of gamma in one or multiple regions, or is it about them becoming aligned? Because I know in humans, well, this is the theta sort, but in humans, theta resets when you enter an encoding state, or at least the evidence is pretty compelling that that's true. Is this something where the gamma is out of alignment, and then when you enter an encoding state, one of them is reset, now it's in sync between C1 and C3?
So the question is, it could be the case that gamma was going on all along, but kind of unsynchronized in different regions of the hippocampus. Is it the fact that there was going on along and then just snapped in together during exploration? Or was it the fact that you were getting a little bit of gamma, and then when the rat started sniffing an object and was going to remember that object, you got gamma and it was locked together? We found the latter rather than the former. There wasn't a lot of low gamma power, consistent low gamma power going on during baseline. Now, that's not to say that it's not there. It just tends to happen in these little sporadic moments here and there that if you look for it, you can find it outside of object exploration. But it becomes really most prominent during novel object exploration. And perhaps other things that we haven't investigated, but that's one way to make it trigger. Annabelle.
So you sort of skipped over exactly how you measured good encoding or bad encoding, but then separated your signals into sort of two categories.
Mm-hmm.
How much did the behavior look like two very distinct categories versus a sort of long schmear and you split it?
It was, the question was there really clearly from a behavioral standpoint an obvious good memory and an obvious bad memory? Or was it a continuum of memory that we did a median split, something like that? It was the latter. We artificially partitioned the performance into these states. And what I can say is that it doesn't matter exactly how you partition them, you still see the general finding. That obviously it's not as if you either get memory or you don't have memory. You have better or worse memory and gradations between. And the only way for us to be able to partition into these states was to pick something artificial. And to reassure ourselves that it wasn't just a quirk of how we partitioned, we picked this first, went with it, but then in our neurotic moments, we did it a couple different ways to make sure that when somebody asks this, we can say, it doesn't matter exactly how we partition it. Great question. All right, so now I'll switch to talk about, in terms of thinking about good memory and poor memory, or something that is memorable versus something that's forgotten. One of the big lessons that we can take from the psychological literature is that emotional memories tend to be remembered better than neutral memories. Not always, and that there's extreme versions that lead to dysfunction such as PTSD, but in general, moderate emotional arousal leads to better memories. And that what we know from lots of work from folks like Jim McGall here is that the amygdala, in particular, the basolateral complex of the amygdala, seems to be a key hub in mediating those emotional arousal influences on memory. And the idea is that you have some experience, and that experience here, a record can be made of that experience in different ways in different locations, but one in terms of declarative memory in the hippocampus and associated structures in the medial temporal lobe in humans. But then there's an opportunity through emotional arousal activating either directly or through peripheral hormones indirectly the amygdala, and then the outputs of the amygdala have the opportunity here shown by McGaw in red to, through neuronal projections, modulate memory processing in other areas so as to be able to prioritize some memories, in this case, those with emotional salience over others. And we were interested in this, but I'll admit that we're less so interested in the emotion part and more so interested in the memory enhancement part. And so an idea that set forward a collection of studies in my lab was the idea that we could directly and at first electrically stimulate the amygdala to try to prioritize memory for some junk neutral, no emotional content objects over others. And so that the basic setup is the same that I've kind of been showing you before. A rat comes up and sniffs a novel object, and then later in the future, we ask that rat to show us, does it remember the object or better? And then one wrinkle is in this line of work is that at the offset of exploration, we tickle the amygdala with a little bit of electrical current. We're talking small, 20 microamps, and you can see it's in a particular pattern here that's kind of was meant to be a guess at mimicking theta modulated gamma. But this is just a small amount of current delivered for 1 second at the offset of exploration of that neutral object. And the question was then, when we brought the rats back later, would they remember the object that previously was followed by amygdala stimulation better than a control object? And we, of course, have to have novel objects here as a reference for asking, just in general, can the rats remember the repeated objects more so than the novel objects? And this is a complicated schematic, and I'm not going to walk you through it, other than to say we didn't just use 3 objects. We used lots of different objects, counterbalanced in different locations, the other part is that after the rats during the study phase, when the stimulation was delivered on after some of the objects, for half of the objects we tested memory immediately, and for the other half, 1 day later. Excuse me. And the point is that the point we did that, and the point that I'm telling you is that even from just looking at Jim McGaw's work, we predicted that the benefit would be most prominent here 1 day later. And in fact, over several experiments, that's exactly what we found. That here in the red, stimulation, and blue, no stimulation on the immediate test, again, this discrimination index where 0.5 is chance and 0.66 would be really good memory, that there was not a difference between the 2 conditions on the immediate test, but then when we looked on the 1 day test, that the objects that were previously followed by amygdala stimulation were remembered as good as the objects that were tested immediately. And in contrast, there was very little memory for the objects that had not been, on that 1 day test, had not been followed on the initial study by amygdala stimulation. And we, in a series of experiments, the first being just, could we get that behavioral effect? Then the next one was, does that depend upon the hippocampus? And for the sake of time, the short answer was using mucimol, yes. That in experiment 3, the question was, what might stimulation of the amygdala, what kind of network state might that induce in the hippocampus? And I'll show you a little bit about that. And I should say that then first, because we're using electrical stimulation, it's always tricky to record electrical activity at the same time. And so for this, we took advantage of the fact that the connectivity from the BLA of the amygdala to the hippocampus is almost exclusively ipsilateral. And so if we're recording from here, that we should expect only artifact from here, and artifact plus actual neuronal something going on here about the one brain area impacting the other. And so just in this little time-locked thing, we do find that there are some small artifacts from the electrical stimulation shown at each pulse here at one of those triangles. And just to basically clean it up, subtracting the two makes this look even better. And so this is a way to help us understand that we're not just recording the electrical artifact of electrical stimulation. And what we found was that stimulation of the ipsilateral amygdala led to a robust increase in CA1, CA3 slow gamma coherence, and not so much on the other side. And you might ask me, well, isn't it obvious that if you stimulate the amygdala somewhere around 50 hertz and you record in the hippocampus and you see something around 50 hertz, isn't that you just puppeting what you want to see? And the answer could be. And we did another condition in which we stimulated the amygdala at 80 hertz, just picking something that's not in the slow gamma range. And we did see that it was able to induce something around 80 hertz, but we still also recorded this increase in the slow gamma range in the hippocampus for the ipsilateral, not for the contralateral. And this to us suggested that there's the possibility that activating outputs from the amygdala, at least within some range, induces the hippocampus to go into whatever its resonance oscillation might be. And at least on one front, that resonance state is that slow gamma state. Now, excitingly enough, or depressing enough, that it's not only the hippocampus that looks the same between you and rats, the amygdala too. And the use of electrical stimulation in our rat studies opened up the possibility, in collaboration with neurosurgeons at Emory, and under the good fortune that patients hanging out in the epilepsy monitoring unit with informed consent volunteered to participate in these studies, we were able to replicate the rat studies, basically doing it exactly the same in humans. And so instead of allowing humans to sniff or whisk junk objects, because, well, you might not know, but you don't have active whisking, and your sense of smell is pretty bad. But in patients with electrodes for clinical reasons implanted into the medial temporal lobe, we're able to target those that happened to be centered around the basolateral complex and deliver that same type of 1 second worth of stimulation that had the same pulse train frequency and the same kind of experiment. Now in the humans, it was seeing pictures of novel objects, and for half of them it was followed by electrical stimulation of the amygdala, and for the other half it was not. And we again tested half immediately and half the next day. And what we found in the humans was basically what we found in rats, that there was not a consistent effect between the stimulation in red and no stimulation in blue on the immediate test, but that there was a very consistent effect across the patients. Despite the overall variability in their memory performance, there was a consistent effect of stimulation leading to improved memory. And to kind of crystallize this a bit better, then this plot just shows this plot, but subtracting the no stimulation from the stimulation. And so 0 means no effective stimulation, and greater than 0 is a memory benefit. And you can see on the one-day test, there were a couple of participants that showed no difference, but on average, there was a reliable, statistically significant effect of stimulation. And it was somewhat specific to that one-day test, just like it was in the rats. Now, one thing that we could do in the humans that we could not do in rats was to ask them, so what'd you feel? So we did. And as a separate little side experiment with every participant, we put the button, and said, okay, I just pushed it. Did you feel something? And in fact, half the time the button we pushed delivered nothing, and half the time it actually delivered the same stimulation that we used in the experiment. We thought that we would need to try to sort out false alarms from real hits. But in fact, in this completely ridiculous graph that I insisted we put in the manuscript because I love it, 100%, all 14 of the patients for all of the trials said, no, I did not feel anything. Because from the beginning that we started this experiment, people were like, oh, but aren't you fear conditioning? Aren't you, amygdala, doesn't that do fear? I'm not saying that's not true, but that the outputs of the amygdala have the capacity to prioritize memory, even if upon direct activation outside the scope of emotional experience, can prioritize memory even when the participant feels nothing. And so that's why I insisted upon this ridiculous figure where there can be no error bars, because all 100% said, no, I can't feel it.
Did they feel other things, not the stimulation itself, but did they self-report any other side effects?
Yeah, so what I should say is that this was done with John Willey and Corey Inman at Emory in a separate paper that Corey did. I mean, this sounds... These are all, what I'm going to say, because it's within the context of patients, that these are all within the healthy, normal ranges of the levels of stimulation. This was tiny, right? But to kind of parallel the rats. They did ask in the patients in a separate experiment, well, what about now? Well, what about now? Well, what about now? Well, what about now? And eventually, if you got it about 5 to 10 times high of a current as we used here, somebody say, I think I felt something. In one patient, that patient said, not in this, way higher levels than we used here, said, I feel terrified like I was ziplining, but only on the left side of my body. I don't know what to make of that, but to make the point that it's not as if it's not as if everybody's wrong, the amygdala has nothing to do with fear. It probably just isn't that conscious appraisal until you ramp up the stimulation so much that you're probably stimulating like neocortex wide kind of phenomenology. Okay, so this is now all three of the studies, the rat studies and the human data plotted on a similar scale to show not only does it look the same, but the effect sizes were so close to 1 in each in a Cohen's D that it was almost too good to be true, and maybe it is. I mean, maybe subsequent experiments will pull the rug. But for now, it looks as if the mechanisms of amygdala prioritization of declarative memory, to the extent that you can find it through electrical stimulation, look pretty similar. Now, then the question is it that the stimulation, activation of the amygdala and the release of glutamate downstream in the hippocampus triggers the hippocampus to go into this oscillatory state of slow gamma, and that's a good memory state, and that promotes synaptic plasticity and synaptic transmission, and it's that mechanistic thing in the hippocampus and related structures that makes good memory. It could be that, or it could be lots of other potential explanations. And we're trying to track that down to ask really, is it something akin to this? And one approach that a graduate student in my lab, Nathan Algram, is following up is to use optogenetic stimulation. Now we're kind of breaking paths with what's possible to do in parallel with the humans, at least now. But when we transfect glutamatergic neurons in the BLA with channel rhodopsin and verify that the terminals are there in the downstream hippocampus, and then shine light and record in both the amygdala and the hippocampus. One question can be, what's the best way to induce oscillations in the hippocampus via amygdala activation? And that this is just don't worry, and little colors, like, whoa, right, that it's a couple of different efforts of picking some types of optogenetic stimulation of the BLA in a control condition in which we shine a light that's outside the spectrum of the opsin that we're using. And essentially what we found is that no matter how you pulse the amygdala, at least within these constraints, will happily bounce along with you. The question then is, to what extent is that optimally or effectively transmitted to downstream hippocampus? And to our surprise is actually that stimulating the amygdala at that theta oscillation isn't very effective at inducing increased power in the theta range in the hippocampus, and not much more so than what should be the control near infrared light. But things with 50 hertz as part of the pulse train do, and we're moving towards trying to understand then what are the kind of more endogenous oscillatory interactions between the amygdala and the hippocampus to try to push that forward to understand using amygdala stimulation as a window into good memory states in the hippocampus. So what I'm going to do is tell you that I'm not going to tell you about this last thing, and that it is that we were interested in for humans at least, when information that was initially formed by declarative memory, one feature of that is that it then is subsequently available to what we often subjectively perceive as conscious recollection. Less theoretically described here as post-retrieval monitoring. And that was an interest to us, and that then gets us into this range of areas and the neocortex that really are different across species. This part, very similar across species, not so much here. And so we wanted to then use scout-based EEG recordings, and this was done in collaboration with my colleague, Patricia Bauer. And that we did an autobiographical memory retrieval. We found alpha oscillations like Berger found, and that's why that's the first, it's alpha. And that we made really neat animations that was going to make you go, wow, and that there were differences between males and females in the extent to which they did post-retrieval monitoring, but you have to go to class. So I'll bring this here to then say what I've done is to set up something that I haven't given you a payoff for. It's like, how is it, why is it, and how is it that I remember that thing, that event with my son out on the beach a year ago, and I imply that that's important to me. But what I have not really said is what is important, what makes a memory worth remembering. And I think that the answer is that that is going to differ. And so I actually was going to bring back another picture of my son to highlight the differences across species in terms of, well, to us, maybe, you know, that beach thing makes sense to remember, but for the tenrec, it's like, where did I find the tasty cricket? And then as searching for my own pictures of my son, I swear I didn't make him pose for this. It occurred to me maybe some of those differences, even at that psychological, phenomenological level, aren't all that different between humans and tree shrews. So I can tell you for sure, even though he doesn't remember Hilton Head, my son definitely remembers where to get the good noodles at the place near Emory's campus. But that, thinking about it, there will be species-specific priorities used to prioritize information. And that I won't, and I can't answer what makes a memory memorable, because that is both species specific and idiosyncratic among us. What I would have said is that there are sex effects even within humans to influence the memorability of different memories. But I'll leave you with these two creatures eating their food to then say, this last part here, that there's lots of, when we think about the neurobiology behind it, this evolutionary tree of mammalian neocortex is to make the point that there's extreme diversification of the neocortex across the diversification of mammalian species. But that the point that keeps bringing me back to study hippocampus-dependent memory is this point, the point that at some level, at something that we can understand at the network, at the computational, algorithmic level, that makes super important differences for how you experience it as a human, as you experience it as a tenrec, but that the neurobiological mechanisms should be conserved across the mammalian taxon. And then the question is, exactly what did evolution stumble upon 200 million years ago that was so good that in the course of the diversification of the mammalian taxon and the mammalian neocortex, every opportunity looked at the hippocampus and said, nah, it looks pretty good. I think I'll keep it there. So that idea, then, it's not about remembering everything. It's some compromise of remembering what's important and having good memory states and poor memory states and the capacity for other structures to intervene. I've talked about the amygdala today to help with that prioritization based upon other things. That's what is intriguing to me and what will continue to push me forward. So with 2 minutes to spare, I'll say thanks for coming and acknowledge the folks, some of who are in the audience, some of the former graduate students whose work I talked about, here are my collaborations for some of the work I talked about. And with that, I'll stop and see if there are any remaining questions. And I know that I'll be hanging out with some of you afterwards as well. But thank you very much. Yeah.
Both the optical and electrical cases, you did for physical and electrical case, you did 40 hertz simulation, and then I think 80 hertz. Solving trained.
The question was about parameters for the electrical simulation, and that there was a condition with 50 hertz and 80 hertz. Both were modulated by an 8 hertz theta. But yeah, go ahead.
Did you also just try a non-periodic form of simulation? So still, let's say, 50 times per second, you're going to have pulse, but just not periodic, kind of randomly interleaved. See if that also been trained.
So the question was, did we try something, an electrical stimulation pattern that did not itself contain periodicity? And for the electrical stimulation, we did not. For the optogenetic stimulation, that I didn't show you this work, we tried 2 things to try to get at that question. And one was random pulses, matching the number of pulses within a second. And that was not as effective at inducing downstream oscillations. And so that could be that you can interpret that in a couple different ways. The other that we tried was constant illumination of the BLA neurons to try to ask just if we just get them to dump glutamate willy-nilly, might just the presence of extra glutamate from those terminals in CA fields be enough to induce that resonance? And no, the answer was no. But there could be other reasons that was uninteresting, like, for example, saturation of the opsins, and so we tried different things that didn't have periodicity, and they weren't as effective at inducing periodicity in the downstream hippocampus. So there are constraints.
So we'll stop it there. Please feel free to talk to Joe afterwards, and let's thank him again for a great speech.
Thanks for having me.